Daniel has sort of covered some of this background for loss of function variants and human knockout genes. I just wanted to take you through a couple of examples that we have driven, where in the first sample we identify initially a gene through GWAS, then pulled out variants. Some of them turned to be loss of function, and we are now in a clinical development with that. It's a gene called DCR3, decoy receptor 3, which is one of the tumor necrosis superfamily receptor genes, which I'll show you. And the other one is around the PCSK9, because as you know, I mean, loss of function in PCSK9 obviously drives down cholesterol. And the industry is all over that today, but we have data from our children's database that there is actually significant, potentially significant elevation in triglycerides actually in these kids, which is not being addressed with therapy. And let me see here. So this is mostly covered through Daniel, so I'm not gonna spend time on it. This is just sort of the rare and orphan disease biobank that we have at CHOP with 22,000 samples, where we have made hundreds of discoveries of variants. Most of the variants, so both in known genes and new novel genes, most of the variants are autosomal recessive loss of function variants, but as you can see, this sort of really crosses essentially any disease entity there is. And just very briefly on this IBD variant that we identified in DCR3, encoded by the TNF-RSFB6 TNF-alpha superfamily receptor gene. This was actually the first locus we identified, driven really by early onset inflammatory bowel disease. The allelic variant associated with the expression of this gene, both in ulcerative colitis and in Crohn's disease, and correlated with the at-risk variant. This is actually DCR3 is a receptor that blocks three separate ligands that are all very pro-inflammatory in nature. TL1A, which is in drug development at Pfizer currently, LIGHT and FAS. And LIGHT is actually sort of the highest affinity receptor, which was sort of the direction we decided to pursue. We sequenced this gene, we identified multiple variants that were enriched, mostly missense variants, but when we tested function of these variants, several of them were actually null, had no function, so DCR3 was not cleaved and secreted, and this was validated in a separate laboratory by Carl Wehr, who actually discovered this pathway many, many years ago at the La Jolla Institute in San Diego. And this was a paper we published in 2013. We subsequently sought after, if you think about it in the context of what happens if you are missing DCR3, LIGHT binds to its receptor, and it's a lymphotoxin B receptor and the herpes virus receptor. And if LIGHT binds to its receptors, it results in inflammation. There's just sort of a schematic of it. And in normal individuals, DCR3, which is cleaved and secreted, buffers this and keeps homeostasis, so there is not an inflammatory state. But if you're missing, DCR3 and there's LIGHT around, it's going to potentially result in chronic inflammation. We actually reached out to a Japanese company, Kyowa Hakonarson, that had antibody against this, and we managed to license this in, and we are now actually, we have an IND, we're in clinical trial, replacing this particular loss of function variant, because it leads to a loss of function of DCR3, but because DCR3 is a gene that buffers and blocks, you can replace it with an antibody. So this is in ongoing clinical trial currently as an example of sort of implementation of therapeutics from this. PCSK9 is another variant, as I mentioned, and you know that gene obviously very well, and its impact, there are three major variants that have been shown to reduce the plasma levels of low-density lipoprotein in particular. The AREC study showed this very, very clearly, and more robustly in African-American people than in white people, actually. We looked at our biobank. Patrick, who will actually present here a little later today, imputed 8,000 samples because we did not have those sequenced at the time, and he was able to identify 2 of the 3 loss of function variants, and 3 of them actually turned out to be already assessed and had been tested, so we can validate the haplotype imputation. The frequency was very comparable to the databases that we have. And here you see this very similar sort of, lowering in the lipid levels of LDL with these variants in these kids. You see that HDL is actually also slightly lowered, which is sort of interesting. It doesn't go in the favorable direction if you have this particular variant, 13.6%. But triglycerides actually went the other way around. So it sort of, went up a bit. It wasn't statistically significant. It was almost 10% increase in triglycerides. But this is of sort of potential concern if you don't address triglycerides and you're directing therapy against lipid lowering with this approach. So the opportunity exists to replace loss of function variants with monoclonal antibodies, as those 2 examples show. There are obviously multiple other genes and hundreds of other loss of function variants that we have. And we just got funding for actually pursuing 500 iPS cells that we are going to use and generate for these monogenic diseases and screen drug against in the future. So these genotype frequencies are very comparable to other databases, but as I said, the potential concern is raised with this elevation of triglycerides in patients who, where therapy would be directed only at the LDL cholesterol with this approach. This is sort of the schematic model for this, but I know we are a little bit behind in time. So this is sort of just a pharmacogenomic model that we applied for the light antibody, and we hope to get a quick win for that. So with that, I'll stop and happy to take one question if there is an alarming one before break. Thank you. I guess not. So
I have to ask one question.
Okay.
So once you find a gene of interest and the amount of biology that you have done, at least, give us a sense of the time frame for that second to last slide before where you had that from gene to therapeutic targets.
About 3 years. Not terrible. I mean, we actually discovered this variant back in 2008, but it was functionally characterized by 13th, and it took a couple of years to get agreements to get access to the drug. And in the meantime, we did cell-based assays, we did some animal work, and we did some independent laboratory work with collaborators. So process probably about 5 years total. Thank you.